Good afternoon, everyone, and I'd like to add my acknowledgement of the Ngunnawal people, of the traditional owners of the land on which we meet, and I'm delighted to be speaking to you today about zero childhood cancer and how we see that powering the Cancer Moonshot. So many of you will be familiar with the challenges of childhood cancer. It's the leading cause of death in Australia, in Australian children, with 3 Australian children dying every week, despite very substantial improvements in outcome over the last decades. There's about 1,000 new diagnoses every year. About 200 children of these will have refractory cancer or will relapse, and most of these will die. And even for those children who survive, the journey is not over, because many of these will have serious life-altering health conditions as a result of the toxicity of the treatments that have been given to save their lives. So we critically need more effective, safer, targeted therapies. So the approach we've taken is personalised or precision medicine, which has changed the model of care from a one-size-fits-all approach where every child that was diagnosed with a particular type of cancer got treated in the same way, to a more patient-centred treatment based on the genetics and the biology of each child's individual cancer. We established the Zero Childhood Cancer Program as a comprehensive integrated approach initially for the children with the highest risk cancers, and by that we mean those who had less than a 30% chance of survival. It involved biopsies of each child's cancer from around Australia, wherever they were, being sent to our laboratories at Children's Cancer Institute for very comprehensive molecular profiling on the left of the slide there, with the idea being that we would identify the molecular drivers in each child's cancer and then find the drugs that targeted those individual genetic changes for that child and making personalised treatment recommendations. In addition, on the right-hand side of the slide there, wherever there was sufficient tissue available, we would also empirically test in the laboratory the response of that child's cells to a range of anti-cancer drugs, either by high-throughput screening of up to 150 compounds safe for use in children, or by developing patient-derived xenografts or personalised avatars where we could test response to various different therapies. And all of that information would come together, be the results discussed at a national molecular tumour board involving all 9 children's treatment centres around the country, and then a zero recommendation report issued. So this is now our National Precision Medicine Program for Children, and importantly, it is the world's most comprehensive national child cancer personalised medicine program. It started as a pilot in 2015, led by Children's Cancer Institute in partnership with the clinicians at the Kids Cancer Centre at Sydney Children's Hospital. There was a pilot study in New South Wales, and then from 2017, a national clinical trial for these high-risk children was rolled out that has involved over 900 children being enrolled. And as you heard from Sophie, as of the end of last year, Zero has now expanded to all children with cancer, regardless of their risk stage or tumour type. And in fact, as of today, over not 1,800 but 1,900 Australian children and young people with cancer have been enrolled on Zero. And this is the first time that precision medicine has been available for every child diagnosed with cancer in any country in the world. The important thing is that we've also been able to show that this personalised, precision-guided treatment approach actually improves clinical outcomes. This slide summarises the results of a paper we were very proud to publish in Nature Medicine, one of the leading journals, just a few weeks ago, describing the outcomes of the first nearly 400 of these high-risk, rare and relapsed undiagnosable patients on Zero. This is the largest study with the longest clinical follow-up of any child cancer precision medicine program in the world. As you can see there, of patients who received their Zero-guided precision-guided treatment, nearly half of the patients who got that treatment recommendation actually received that treatment. That's the highest uptake of any program in the world. over half of these critically ill patients showed an objective clinical response, a stable disease, complete remission or partial remission that lasted more than 6 months. And in terms of longest survival, precision-guided treatment more than doubled the 2-year progression-free survival in these children. And if you compared progression-free survival in children that got precision-guided therapy or just were enrolled on clinical trials with novel molecular targeted drugs, over a 5 times increase in survival. And then importantly, where precision-guided treatment was given early prior to relapse, a very substantial improvement in clinical outcome, 40% 2-year progression-free survival and over 50% overall survival at 2 years of these children who in most cases were pretty much destined to die of their disease. So Zero has changed the model of care for children in Australia with high-risk cancer. It is now standard of care for these kids. As of the end of last year, our current enrolment figures show that we will be capturing almost all of these children, with over 900 predicted to enrol this year. This approach of using deep whole genome sequencing has been adopted by national precision medicine programs around the world. It's led to more than 20 clinical trials, a raft of high-impact research publications, and importantly, internationally unique data sets linked to clinical outcomes, and also well over 100 of these patient avatar models that are a resource for research not only in Sydney or Australia, but around the world. So where to from here? Clearly, survival rates have improved significantly through Zero, but are they good enough yet? You saw the numbers. There's still way too many of these children dying. So what are the next innovations as part of the moonshot to achieve our collective goal of one day 0 children dying of cancer? So these are our moonshot goals. Firstly, the 02 clinical trial, which is the one that's currently being rolled out to all children with cancer, which is to be understanding the power of this comprehensive sequencing, not just for the high-risk children, but to medium and standard risk patients. We hope that we will continue to have the funding to roll that out beyond next year, and that we can find the right test for the right patient at the right time for every single child in their cancer journey. I'll talk to you about 4 other goals. Liquid biopsy, using a simple blood test to monitor tumour burden and progression in children. Preclinical testing, improving that so that we can increase the number of patients who get a personalised treatment recommendation. Cancer risk, can we use the sequencing of the whole of the normal germline DNA? We do whole genome sequencing for every patient in Zero. Can we use that to identify, monitor, and improve outcomes of patients and families at risk of developing cancer? And can we increase treatment options for these patients by improved drug access and new drug development? So firstly, liquid biopsy. We know that a single surgical biopsy is unlikely to capture the true molecular diversity of a tumour, and that different regions of the tumour can have different cancer cells with genetic changes growing in them. And yet we make treatment recommendations based on the bulk analysis of that single tumour biopsy, knowing that there may be key targetable genetic changes in other parts of the tumour that we're missing. We also know that through the cancer journey, the cancer mutates and changes in response to treatment, and that resistance or responsiveness to particular drugs may be determined by the emergence of cancer cells that have changed throughout that journey. So ideally, you'd be doing multiple biopsies, but you can't do that either ethically or practically. But can we use a simple blood test to give us this information? And our data suggests that we absolutely can. This is just the proof of principle. On the left-hand side there, that's the genomic data of a single child with high-risk neuroblastoma. And you can see there in green, genetic gains, and in red, losses. This is all the chromosomes of that child. This is the DNA in the blood of that same child, and you can see these same genetic gains and losses reflected because there is circulating DNA from that tumour in the blood of that child. Now, this was done with a technique called low-pass whole genome sequencing, which is great, but we know it's not sensitive to be able to make detailed, personalised treatment recommendations. So what our team has now done is to develop an approach for the rapid construction of personalised panels for each patient based on the initial whole genome sequencing that's done at the time of enrolment on Zero. This test has been applied to now nearly 170 patients using peripheral blood or also cerebrospinal fluid for brain cancer patients. And we've been able to show that it has the sensitivity and the specificity in the blood to measure tumour burden, to identify these new genetic changes in the tumour as they're emerging, and to be able to then make personalised treatment recommendations based on the genetic changes that we can see in the blood because they're a mirror of what's going on in the tumour. And that provides the opportunity to intervene with novel therapies before the patient has clinically relapsed. And we know from years of our research with acute leukemia that if you do that, you intervene based on molecular data before relapse, you can significantly improve outcomes. And we've shown that this can be done with samples from all over Australia, again, potentially changing the model of care. Imagine you've got a child in a rural country town wondering whether their tumour has progressed. Instead of having to come into a children's hospital for a CT or an MRI, their GP could take a blood test, send it to our laboratories. We do the test. Faster, cheaper, quicker. We can provide the information that the big scans can't because we can get this data so much earlier. Let me move now to preclinical drug testing. I mentioned that we do high-throughput drug screening and we create patient avatars. Why is this important? Because our Zero data has shown to us that 30% of all the children on Zero, 30% of these high-risk children, don't actually have in their tumour DNA a genetically actionable change. That is, we can't make a personalised treatment recommendation for 3 out of 10 of these children. For these patients, getting that empirical evidence of what that tumour cell, what their tumour cells, which drugs they will respond to, is their only way of getting a personalised treatment recommendation. And so we've published papers already showing that Zero preclinical testing can lead to identification of new therapeutic options and prediction of drug responses in 2/3 of patients. We've shown that there is a strong correlation between the clinical response and the responses we see in the high-throughput screening and in these xenograft models. And we've shown, importantly, that these preclinical models can check for sensitivity not just for individual drugs, but for drug combinations as well. So clearly, integrating high-throughput drug screening with molecular profiling and precision medicine can maximise personalised therapeutic recommendations. But the gold standard of this testing is testing in these avatar models. But it's slow, it's expensive, and it requires very specialised skills. How can we do that better? So we've partnered with our colleagues in the Canadian Child Cancer Precision Medicine Program to use genetically modified fish that are transparent. And you can inject the cells, which have been fluorescently labelled, into the yolk sac. You can see the cells there. You give them chemotherapy, you can see that the number of cells has shrunk. So here's data from just one patient demonstrating proof of principle. Can I go backwards? What have I done? Yep, sorry. There we've got a patient that responded to these treatments with Ewing sarcoma. We were able to model that in the mice. There's the complete responses in the mouse as well. Took months. So this patient responded to this, did not respond to this treatment. This is the data in the zebrafish model. That's day 1. Each of these dots is an individual zebrafish. That's day 4. The cells have increased. So anything above the line means that the tumour cell number is increasing. They're not responding to therapy. Anything below the line means that they're responding to therapy. These are the 2 treatments that the patient responded to. These are other treatments predicted to work. All of these treatments above the line show no response, including the one that the patient didn't respond to. So we've now done this in 10 patients across a whole range of different tumour types. We can see a very strong correlation between the larval zebrafish and the mouse zebrafish responses, very strong correlation with the exception of one patient of all the patient responses, and we've been able to do this in 1 week instead of the months that it takes to do this in mouse models. So this is the sort of innovation that we can bring to these programs to bring more treatment recommendations for children. Turning to cancer predisposition, we know that a significant proportion of children are born with a genetic risk of developing cancer. And it's important to know that because it can improve treatment and enable early detection. So our zero germline testing has found that 1 in 6 children with high-risk cancer, and germline is just the normal DNA, are born with a genetic cancer risk. That's roughly 150 children a year, and that's based on the cancer predisposition genes that we know. And through our research, we know that we're likely to find many, many more of these. And importantly, fully half of zero's risk finding in children would have been missed by current standard of care recommendations for genetic testing. Here's just a couple of examples about how knowing what's going on in the germline can affect treatment. So here's a 16-year-old boy with sarcoma. Zero testing found a variant in a particular gene, TP53. Wouldn't have found that by current testing. Based on that finding, the young man's management was changed, radiotherapy was reduced, and increased surveillance has been introduced for that patient because we know that in the disease that comes from this type of germline variant, you can improve survival significantly by surveillance. Can also help relatives. This is a 5-year-old boy with a brain tumour. Zero identified an inherited risk in a predisposition gene, PALB2. Because of this finding, the family was tested. A female relative was found to share this gene variant. She went for a mammogram, and breast cancer was found early, well before it had metastasized, improving her likelihood of a good outcome. So what are the remaining challenges then? There are a lot of questions that we don't have the answers to. How do our genes contribute to our risk of childhood cancer? How likely is it that a given gene variant is going to cause cancer in that child? How do we identify these children? And how do we convert findings of potential risk or potential new variants into a definitive diagnosis? So there are 2 things that we're doing. One is that we've established a dedicated cancer predisposition program at Children's Cancer Institute, and the other is that through Zero we're establishing a national child cancer predisposition registry, the first for the country. And the moonshot here is that we believe that one day childhood cancer can be prevented with a national registry, this unique zero data, unique laboratory models that we've established, all these experts working together. We believe that we can have interventions to reduce cancer risk and preventing childhood cancer before it starts. We're working on defining new cancer risk genes by modelling them in the laboratory. We're looking at defining better measures of risk by actually having biomarkers in the blood of major child cancer predisposition syndromes. We're using this information to improve surveillance and screening, and we're also developing targeted drugs to prevent potentially certain childhood cancers, sparing them of this entire cancer journey. So we can identify these genetic drivers. What are the remaining challenges to treating these children? Number 1, access to existing drugs. There are too few industry-sponsored paediatric trials for all of the reasons that you see there, and still not enough dedicated funding to support investigator-initiated or international consortium trials. And so many times in Zero, we see targets identified, but there are no drugs available to treat them. So we need new drug development, but it's got to be in academia, in research laboratories, because pharma does not have an appetite for making drugs for children because the market is not big enough. So what are we doing to improve existing access for drugs with cancer? Firstly, we're partnering through the Prospect Initiative, which you've heard referred to, that David Thomas is going to be talking about later. We're partnering with Omico to increase the attractiveness of Australia to global pharma, to run their clinical trials here because of the capabilities that we've established for this comprehensive national sequencing programs from children all the way through to old age. We're partnering with national clinical networks and ANSCHOG to support new investigator-initiated or international collaborative trials. And we're developing new companion trials to zero. This one, for example, Optimise, in partnership with our clinical colleagues, providing access in Australia's first child cancer basket trial to new combination therapies. That's what we're doing. What can government do? It can review legislative and regulatory provisions to encourage earlier and easier access to novel drugs for paediatric cancer patients, such as these regulations and legislations exist in the US, they exist in Europe, they don't exist in Australia. So there's work to be done there. And the other thing that we're doing is that we have established a globally unique paediatric-specific drug discovery pipeline, making drugs, targeting new targets that have been developed through zero. So we have the research groups, we have the dedicated drug discovery expertise, we have the clinically relevant biological models, and there's this national network so that we can make more of these drugs to target children's cancers. So we believe that these are 5 different ways of addressing the Moonshot program. We also see it being powered. You heard Dorothy talk about the Australian Comprehensive Cancer Network. We're very proud that next year we're going to be moving into a brand new building, the clinicians at the Kids Cancer Centre and our researchers at Children's Cancer Institute, into Australia's first Children's Comprehensive Cancer Centre, which has capacity for 900 child cancer clinicians, scientists, doctors, allied healthcare workers. And we see that as driving that cancer moonshot forward. And I'd like to finish it by acknowledging this huge team of people, doctors, scientists, families, parents, patients, who've helped power this work so far, and acknowledging all of the support for our program. Thank you.